Alright, good day everyone. Welcome back to my channel once again. I still remember my humble self, MLS Josiah. Here this afternoon, we'll be running a test known as DNA extraction using magnetic bead method. I will be showing you guys each of the reagents we are going to use for this extraction method. First reagent is known as proteinase K, followed by lysis buffer, you can see, followed by magnetic beads reagents, followed by internal control. You must add internal control across every extraction that you want to do, followed by washing buffer 1. There's washing buffer 2. Here is our sample, and the last but not the least is elution buffer. Here is our centrifuge. You centrifuge each of the step from here. After here, you centrifuge before adding this one. You centrifuge before adding this one, and then here is our vortex mixer. We will be using it to be mixing in each of the steps. Follow the manufacturer instruction. This is our vortex mixer. Here, these are the tips. The tips that you'll be using in this our pipette. Okay, these automatic pipettes. Okay. These are the pipettes, and here are the tips. At some certain point you incubate at 60, and at some certain point you incubate at 80 degrees centigrade.
Before you run PCR tests, you must be properly dressed. Wear your hand glove, your lab coat, and make sure you pay attention to every protocol. Okay. Okay, 20 macroliter of proteinase K to our tube. Okay, that's 20 macroliter of proteinase K. Okay, 20 macroliter of proteinase K is an enzyme. You can see we are adding 400 macroliter of lysis buffer. 400 macroliter of lysis buffer. So you can see 400 macroliter of lysis buffer. You add it to the proteinase K in the tube. We will mix very well, because the next one is our magnetic bead reagent. Okay, you add 100 macroliter of our magnetic bead reagents to the tubes. You mix. You are adding 1 macroliter of internal control to the tubes, to the both tubes. Add 1 macroliter of our internal control. And this internal control must be added across every extraction that you must do, for it helps to know during amplification. It will help us to know if the extraction has been done properly. You can tell during extraction. Okay, we are adding 300 macroliter of sample. 300 macroliter each of the sample to our reagents. Okay, after that you mix. After mixing, you incubate at 60 degrees centigrade using water bath for 10 minutes, and you are to mix 5 times during this process. Okay, you have to mix 5 times during this process. This process enhances the breaking of cells to release the DNA, because what we are interested in is the DNA, and this DNA is contained inside a cell. So the reagents alongside with this incubation will enhance the breaking of this cell, the lysing of these cells. It will speed up the reaction, thereby breaking the cells to release the DNA. Now when you bring it out, you mix. As you can see, we are mixing. You incubate, you centrifuge briefly. After centrifugation, you place the centrifuge tubes on the magnetic tray. You discover that the magnetic bead contains the DNA, will adhere to the magnetic tray. Then you suck out the excess fluid. You add our washing buffer 1, 900 microliter of washing buffer 1. You mix by inversion for 2 minutes. You centrifuge briefly. So you can see we are centrifuging. It's done. You place it on the magnetic tray. They'll get adhered to the magnetic. You suck out the buffer. Suck out the buffer. In this step now, we are trying to wash the cell. That is what it means. We are washing the cells to get a purified form of DNA. We don't want any debris. We don't want any impurity. Look at the magnetic beads contains the DNA. Get attached to that side. Now we are adding washing buffer 2 now. Adding washing buffer 2. Washing buffer 2, as you can see. 900 microliter of washing buffer 2. You mix by inversion for 1 minute. You centrifuge briefly. The essence of this process is to wash the cell, okay, to get a purified form of DNA. You place it on our magnetic tray. You suck out the excess buffer. As you can see, contains the DNA. Get attached to the magnetic beads. Now we are adding elution buffer. We are adding elution buffer. This elution buffer, as the name signifies, it helps to release the DNA that get attached to the magnetic beads. Okay, it helps to elute, to release the DNA. Okay, to detach the DNA from the magnetic beads. So after that, after you mix, you place it in water bath at a temperature of 80 degrees centigrade for 5 minutes, and you mix 2 times during the process. After that, you centrifuge. After you centrifuge, you place it in our magnetic stand. Then after that, you can suck out your DNA and transfer to a new tube. What you can see there is our DNA now. That is what we are going to use. This is our DNA now. This is what we are going to use to amplify and detect using real-time PCR machine to know the load of this virus in the body, in the system of an individual. You can see the magnetic bead now that adhere to that one is useless. We are no longer needing it. Thank you guys. If you are watching this video for the first time, don't forget to subscribe to my YouTube channel, comment, reshare and like. Thank you.